morning, dear students. In the last video, we have learned about the two DNA repair mechanism under direct DNA repair. In today's section, we are going to see another type of DNA repair, that is methyl mismatch repair. It is otherwise called MMR. Okay. And we will discuss about the process of this type of DNA repair in the case of prokaryotes and eukaryotes in this section. So, in which all occasion, which all situation this methyl mismatch repair function, that we need to understand first. Whenever some unwanted bases got incorporated into the daughter strand by the DNA polymerase during DNA replication, then that time what we lack, this methyl mismatch repair act to correct that error. So, which are all those unwanted bases that we need to understand? First one, uracil. We know that uracil is an abnormal base in DNA. Usually, uracil is present in RNA. So, how uracil can come in DNA? Because of the deamination of cytosine. Okay, deamination of cytosine change that cytosine to uracil. So, uracil become an unusual, unwanted base in the DNA. Its presence that trigger the methyl mismatch repair. That's one case. Another case, adenines deamination. Deamination of adenine form what? Hypoxanthine. So, that also an unwanted base. So, uracil, cytosines deamination, then adenines deamination. That also both these situation results in the presence of unwanted bases in the DNA. Then another case, so many different types of methylations. Methylations of normal bases also resulted in the formation of unwanted bases. So, all these situations need to be corrected. We know that what will happen. I already told you a similar situation in the case of DNA direct repair. Whenever the cytosine get modified to uracil, normally we know cytosine is pairing with guanine. As it is modified to uracil, it only pairs with adenine, and by that way that abnormality stay there, sustain there in the DNA, and it will continue over several rounds of replication. So that type of alterations are very deleterious. We need to avoid that, to correct that. So let's see what is the process, how methyl mismatch repair function. It works in the site of error. Before that, one thing we need to understand. So I'm going to draw a DNA strand, and this one is the template strand, and in the template strand, wherever this type of sequence present, what type of sequence? GATC sequence present. In this sequence, wherever adenine is located, it will get methylated, and that methylation acts as a label to show that this is the template parent strand. It is a label to indicate that this is the parent template strand. And this type of labeling only present in the case of prokaryotes. We are discussing the mismatch repair in the case of prokaryotes first. So, in the case of prokaryotes, how the template parent strand is identified. because of the presence of methyl group that got added to the adenine at points of sequence, which sequence GATC sequence. And which enzyme is doing this methylation? It is done by deoxyadenine methylase, otherwise called DA. So this methylation is done by deoxyadenine methylase or DA. Okay, then we are going to see a mismatch here. So we know that guanine need to pair with cytosine, but as it is deaminated, it changed to uracil. So this is a mismatch error. And the presence of mismatch, it initiate the methyl mismatch repair, and in that repair process, which is the first protein arrived at point mute S, which is a protein mute S. We need to note that protein mutus. And this mutus didn't have the capability to discriminate between parent strand and the daughter strand. Okay. And this mutus then recruit another protein, that is mutal, where it comes and bind to those methylated portion near the mismatch. Okay. So, by that way, it understand or it recognize the parent template strand. And what this mutal is doing here? This mutal prepare the daughter strand, especially at the point of mismatch, to get repaired by forming a loop there. So it forms, this mutal forms a loop here. Okay, still we should know that there is hydrogen bond that is holding this base with this one. And this mutal then recruit another protein, that is mutach. It has endonuclease activity. It has endonuclease activity, and it make two cuts on either side of the mismatch. And after that, all these proteins leave. So now we can see two cuts. And still, we know the hydrogen bond is holding that the incorrect base in position. And we need to dissolve those hydrogen bond. For that, there come another protein, UVRD. This UVRD, it removes the hydrogen bond that holds that wrong missed or that damaged portion of DNA with the parent template strand. Okay. So by that way, after removing that damaged portion, there left one gap, and that gap will be repaired by which enzyme? We know which is the enzyme that is doing the synthesis of daughter strand or the polymerization of nucleotide in the case of prokaryotes. Yes, that DNA polymerase 1. That DNA polymerase 1 came and add nucleotides to the 3-OH end. So it add, and after completing the addition of nucleotide, there will left one nick. Nick is what? What is the difference between a nick and a gap? Gap means there is enough space to add one more nucleotide. Nick means there is no sufficient place or space for adding any more nucleotide. Only possible to form a phosphodiester bond. So that gap, because of the nick, here is sealed by another enzyme. We know its name. What's that? Yes, DNA ligase. It sealed that gap, and by that way repair the damaged DNA to a normal one. This is the process in the case of prokaryotes. Then what is the difference? What are the other ends or proteins involved in this methyl mismatch repair in the case of eukaryotes? Let's see that in the case of eukaryotes mismatch repair, there present a complex of protein and two among them is MSH2 and MSH6 protein. And these proteins, they are analogous or similar to mute S. We know that this mute S is coming and binding to the means damaged site or the mismatched site first. And here this MSH2, MSH6 protein complex can discriminate. It can discriminate between template and daughter strand. But in the case of prokaryote, that mutex cannot discriminate between the template and daughter. Here it can discriminate. How? By locating the position of PCNA. PCNA we know is a clamp loader in the case of eukaryotes that we have learned in the case of DNA replication in eukaryotes. So this PCNA, by locating the position of PCNA as well as the 5' end of Okazaki fragments in the daughter strand, this MSH2 and MSH6 protein recognize between template and daughter strand. Then there is that binding of MSH2 and MSH6 protein to the damaged site trigger binding of another protein, two protein, MLH1. It has endonuclease activity and another one PMS2. And these are analogous to mutel of that of prokaryotes. And what it is doing there? It introduces a cut on either side of the mismatch. Here also it is doing the same thing. Endonuclease cut the newly synthesized daughter strand in its DNA backbone. Okay. Then DNA helicase. There UVRD. Here some other helicase came and unwind or dissolve the hydrogen bond holding the damaged portion of daughter strand with the parent template strand. Clear? Suppose this is the parent strand and this is the daughter strand, and where that mismatch present, that hydrogen bond, it is removed by the DNA helicase. And by that way, it is completely free to remove from here. And in the case of eukaryotes, they present another enzyme that is an exonuclease. Its name is exo1. It excises and disintegrates the nucleotides of this fragment and creates a gap here. Okay. And after that, this exo1 leave that site. Then in the case of eukaryotes, we know that DNA pol delta is the main enzyme to add nucleotides, and it came and filled the gap. And there also, after filling the gap, there left a nick, and that will be sealed by DNA ligase. DNA ligase. So that is the process in the case of eukaryotes. I hope that the entire process of methyl mismatch repair is clear to you. Why that process is called methyl mismatch repair? Because it recognizes that mismatch based on the methylation of the parent strand. Looking to that methylation, it discriminate between parent and daughter strand. So maybe because of that DNA repair mechanism is known as methyl mismatch repair. Okay. With this, I am going to wind up today's section. Thank you.
should fuck her in a Benz.